Gardnerella vaginalis is a naturally occurring bacteria that exists in the vagina. However, in too dense of populations, it can lead to a condition called bacterial vaginosis. This bacteria is also connected to the cosmetic industry and standards imposed on women in society. To give a brief history, this bacteria was discovered by scientist Sidney Leopold in 1953. And later in 1955, it was further worked on by scientists Gardner and Dukes. And this is where the name for the genus Gardnerella came from, to describe this bacteria, as it could not be classified into any existing genuses at the time due to its characteristics, including its cocobacillus shape, its immotility, that it is gram-positive. However, it is considered gram-variable because in certain visualizations it appears gram-negative due to its extremely thin cell walls. It's also catalase negative. It's selective for blood agar, and it is a facultative anaerobe as well as an acidophile. With this in mind, the Gardnerella vaginalis thrives in the vaginal microbiota due to these characteristics, as the vagina is a highly acidic environment in order to kill off pathogens trying to enter, and it is also a relatively anaerobic environment because compared to other parts of the body, a blood supply to the vagina is relatively limited. However, where it becomes a problem is when Gardnerella vaginalis starts to dominate the microbiota, because typically the vaginal microbiota is dominated by the bacteria lactobacillus, specifically 4 different species of lactobacillus, and then features Gardnerella vaginalis and other anaerobic bacteria in smaller quantities. However, when there is too much GV, this can lead to bacterial vaginosis. And due to this, the pathogenicity of GV has been debated for a long time because it does not satisfy Koch's first postulate, which states that a pathogen must be present in unhealthy individuals and that it must not be present in healthy individuals. Moreover, there are 3 main symptoms of bacterial vaginosis, including a vaginal pH greater than 4.5, a fishy odor, and vaginal epithelial cells covered in bacteria, also known as clue cells. This becomes a problem because when there is an overdensity of GV cells, this disrupts the typical metabolic processes of the vagina, such as breaking down pathogens trying to enter, and how the vagina receives nutrients, such as oxygen and glucose, which typically diffuse through the submucosal layer of the vagina, as the vagina, like the GI tract or like our throat, is covered in a mucosal layer. However, the mucosal layer of the vagina is lined with epithelial cells. This brings us to the causes of bacterial vaginosis. It is not a sexually transmitted infection. In contrast, it is caused by a variety of internal and external factors, including chronic stress, changes in hormones, as well as sexual activity, or beauty products, douching, anything that can introduce foreign chemicals into the vaginal microbiota, which is pretty sensitive because in other areas of the body, in the biome, diversity is favorable. However, in the vagina, this is not so. Diversity is not favorable. Something else that influences this is the beauty industry, which historically has targeted marginalized groups of people. For instance, women, in this instance, specifically women of color, by creating an insecurity in them through advertisements and marketing strategies. In this instance, an insecurity such as a vaginal odor, and then providing them with an antidote to this imagined insecurity that was created. Whereas, unfortunately, these products that are being sold, and such as vaginal douches and wipes and sprays being said that are going to correct this vaginal odor, are actually more harmful as they introduce harmful chemicals in the vaginal microbiota, and can just cause disruption in the normal metabolic processes and lead to things such as bacterial vaginosis and all of the other health issues that are associated with it. Unfortunately, 23 to 29% of women annually experience bacterial vaginosis, and about $4.8 billion is spent on treatment every year, according to a journal titled The Female Vaginal Microbiome in Health and Bacterial Vaginosis by Shen X et al. From 2021. All things considered, there are a couple of tests to confirm bacterial vaginosis if it's suspected in a patient. These tests involve a vaginal culture being taken and this culture being sent to the lab to look for milky white fluid, clue cells, or to examine the makeup of lactobacillus, GV, and other bacteria in the vagina by looking at this culture. And treatment is typically oral antibiotics or an intravaginal gel. These treatments are cost-effective and produce results pretty quickly. However, there is a 50 to 67% chance of relapse within 6 to 12 months of completion of treatment of these. And BV is also associated with other conditions such as infertility, preterm birth, postpartum endometriosis, pelvic inflammatory disease, HIV, and human papillovirus, HPV, the most common STI in the United States. Furthermore, the reason for this recurrence is related to GV's cells' tendency to form biofilms, which form around these GV cells in the vaginal epithelium and protect them from antibiotics and bacteria. However, there are new advancements being worked on, according to Shet X et al. 2021, using enzymes such as DNase and lysozyme, which have been shown to break down biofilms and prevent their formation. These used in conjunction with antibiotics can hopefully not only restore the balance of the vagina by repopulating lactobacillus and killing off GV cells, but also by preventing relapse. And in conclusion, further research and increased awareness of GV is imperative, as this bacteria has many implications for women's health and well-being. Thank you.